Hello my dear friends, welcome back to CSIR at home. In this video, we can study about biochemical characterization of Escherichia coli. In the previous video, we have studied about water quality analysis. We have studied two methods, which are the ambient method and membrane filter technique. Okay, for example, if you are getting one water sample for analysis, what do? You either go for ambient method or membrane filter technique. And what is your result? You got, if your sample is positive for Escherichia coli or any other coliform bacteria, okay, you need to do further test. Okay, so in the ambient test, which are the three different methods, three different steps: presumptive test, confirmed test, and completed test. What was the last step in that? We streaked the culture which was positive. We streaked onto the eosin methylene blue agar. And what was the result? The colonies, if it is positive for coliform bacteria, the colonies will be appear as green metallic sheen. Okay, colonies with green metallic sheen. That is known as presumptive Escherichia coli like colonies. Okay, so what we need to do next? We need to confirm it as Escherichia coli by either biochemical characterization or any molecular techniques. Molecular identification of any genes, for example, UID gene or MDH, etc. that we can study later. So, in this video, we can study about biochemical characterization of Escherichia coli. Okay. So, we can study about IMViC test. Okay. IMViC test. IMViC test means indole. I stand for indole. M for methyl red. Okay, here you can see I stand for indole. IMViC test. I stand for indole. M for methyl red. V for Vox-Prosker. Okay, and this I is for ease of pronunciation. And C for citrate. IMViC test means indole, methyl red, Vox-Prosker, and citrate. Okay. The identification of enteric bacteria is very important in determining food or waterborne diseases. Okay, how we are identifying? Using biochemical test or molecular biology test that we discussed. That the most of the enteric bacteria belong to which family? Enterobacteriaceae. Okay, most of the enteric bacteria belong to the family Enterobacteriaceae. It's very important. Okay. Most of the enterobacteria belong to which family? Enterobacteria. And these bacteria are gram-negative, non-spore forming bacilli. Okay. The enteric bacteria are gram-negative and non-spore forming bacilli. And these bacteria we can subdivide it into two classes. That is lactose fermentase and non-fermentase. The bacteria which can ferment the lactose present in the medium is known as lactose fermentase. And non-fermentase means they do not ferment lactose. Now we can study the examples of bacteria which ferment lactose. The examples are Klebsiella, Escherichia coli, and Enterobacter. These are the examples of lactose fermentase. And non-fermentase, which are the example? Salmonella, Shigella, and Proteus. Okay, these are the two classifications: lactose fermentase and non-lactose fermentase. Okay. Now we can study the IMViC test. From the water sample, we isolated the typical E. coli-like colonies. Okay, this is typical E. coli-like colonies from which agar? EMB agar. EMB agar means eosin methylene blue agar. That is specific for Escherichia coli. Then we can subject to IMViC test. Okay, I for indole. So the basis, the principle is indole production in tryptone broth. That we can study in detail. Indole production in tryptone broth. That means the broth we are using for the growth of Escherichia coli contains tryptone. Okay. Then the indole test detect the production of indole from amino acid tryptophan. That is the principle. So we have studied about tryptophan. Okay, tryptophan is a which amino acid? It is a heterocyclic amino acid. Then it is an essential amino acid. Then tryptophan is also an aromatic amino acid. And which is the group present in tryptophan? Indole. Okay. This is the basis of indole test. Okay. Indole production in tryptophan broth. Indole test detect the production of indole from amino acid tryptophan. Then the second test. This is the first test, indole test. Then second test is methyl red. Methyl red. Methyl red is a pH indicator. Okay. It determines whether the bacterium carries out mixed acid fermentation or not. Okay. That is the basis of methyl red test. That is the ability. The methyl red test is assess the ability of the bacteria to produce various organic acid from mixed acid fermentation of dextrose. So, in this methyl red test, which is the medium used, dextrose. The medium contains dextrose. In tryptophan broth, it contains tryptophan. Okay. So, likewise, here the medium contains dextrose. So, the bacteria, if it is positive for methyl red, that means it is able to produce various organic acid from dextrose. Understood? Then the third test is VP, that is Vox-Proskauer test. That is this VP test detect acetoin production. It is a byproduct, that is non-acidic byproduct. Citrate C, that is citrate test determines whether or not the bacterium can use sodium citrate as a sole source of carbon. That means in citrate test we are providing the medium contains citrate as a source of carbon. Okay. The cultures that give plus plus minus minus reaction is confirmed as E. coli. That means plus plus means it is positive for which is the first test? Indole. For indole it is positive, that is plus. Then next test, indole, IMViC, methyl red. It is positive. Then the third test, VP, negative. Okay, E. coli is negative for VP. And last test, citrate. E. coli is also negative for citrate test. That means E. coli gives plus plus minus minus for IMViC test. Now we can study the details of this test. More details. Which is the first test? Indole test. IMViC, I stand for indole test. First, we got E. coli. Okay, on EMB plate. So, we just take that colony, single colony, and inoculate into the tryptophan broth. E. coli is inoculated into tryptophan broth. Okay, and incubate at 37 degrees Celsius. E. coli is inoculated into the tryptophan broth and incubation temperature at 37 degrees Celsius for 48 hours. Okay, after incubation, that is after 48 hours, we are adding Kovacs reagent. Which reagent we are adding? Kovacs reagent into the broth. Okay, into the overnight broth, we are adding Kovacs reagent. And the development of red ring, this is red ring. Okay, the development of red ring in the reagent layer is considered as indole positive. If it is yellow ring, that we can call it as indole negative. Okay, if it is yellow, that means indole negative. What is the principle behind this reaction? Tryptophan broth contains tryptophan. Tryptophan broth contains tryptophan. Okay. And tryptophan, you know, it is an aromatic amino acid. Tryptophan is an aromatic amino acid. And which is the group present in tryptophan? Okay, we have discussed in the previous video. You remember which is the group present in tryptophan? Indole group. Okay, indole group. Tryptophan contains indole group. That is the basis of this test. Most of the bacteria contains tryptophanase. That is an enzyme. Tryptophanase. Tryptophanase is an enzyme. The tryptophanase hydrolyzes this hydrolyzed tryptophan into indole, pyruvic acid and ammonia. Understood? The bacteria that utilize pyruvic acid and ammonia for their energy. Okay, for their growth, the bacteria will utilize pyruvic acid and ammonia. That means the Escherichia coli will utilize pyruvic acid and ammonia. And what is remaining? Indole. That is remaining in the medium. Okay. Then we can check whether indole present or not by adding which reagent? Kovacs reagent. Understood? Kovacs reagent. The Kovacs reagent react with indole to form rosindole. Rosindole dye. Okay, the Kovacs reagent which react with indole to form rosindole dye. That is the bright red compound here. Okay, this is the rosindole dye. Rosindole dye. Understood? That is the bright red color. Okay, that means if Escherichia coli that cannot utilize indole for their energy source. So the medium contains indole. So if we add Kovacs reagent into the medium, it will react with indole to form rosindole dye. That is the basis of indole test. Understood? Next we can study which is the next test. Indole, then M. M for methyl red. Methyl red test. Which is the medium we are used? Here we can use MRVP medium. This medium is common for methyl red test and VP test. Okay, Vox-Proskop test. For both tests, which medium is used? MRVP medium. And which medium is used for indole test? Tryptone broth. Okay. Here, MRVP medium contains dextrose. Dextrose. Understood? First, we can inoculate the E. coli, Escherichia coli, into MRVP medium. Then, incubate at 37 degrees Celsius for 48 hours. Then, after that, we are adding methyl red. Methyl red. That is MR, methyl red reagent. So, after adding the methyl red into the broth, into the overnight broth, what happens if it is positive? Okay, if it is positive, that means red color. Okay, if broth is turned into red, that means that it is positive. That means if it is appears a yellow color, that means it is negative. That is the basis of MR test. Now we can study more details about MR test. The broth we are used MRVP medium. MRVP medium, it contains dextrose phosphate. Most of the bacteria can catabolize which one? Glucose for its energy need. Okay, for its energy need. So bacteria use different enzymatic pathways. Okay, different enzymatic pathways for the catabolism of glucose. It will depend upon the bacteria. Okay, for the catabolism of glucose, the bacteria will utilize different pathways. Understood? So, Escherichia coli will produce different acids as a result of mixed fermentation, including pyruvic acid, then which one? Lactic acid and formic acid. So Escherichia coli is a mixed fermented. Okay, it releases pyruvic acid, lactic acid and formic acid as a result of mixed fermentation. So here the pH will drop into 4. Okay, pH is 4. Okay, after the mixed fermentation in this medium, here, this medium, the pH will be 4. So that's why if we add methyl red, it will turn into red in color. Here, it is not fermented. Non-fermentase, fermentase. That means pH is 6. That's why it turned into yellow color. Here pH is 6, here pH is 4. Understood? That is the basis of methyl red test. Okay, so that means methyl red is a pH indicator. Methyl red is a pH indicator. So that means Escherichia coli utilize the dextrose present in the dextrose phosphate broth and produce different type of acids including pyruvic acid, lactic acid and formic acid. So as a result, the pH of the medium is reduced to 4. pH is 4. That's why if we add the methyl red as drop into the broth, the color will appear as, what will be the color? Red in color. Okay, that is the positive result. Then what is the negative result? Yellow. That means the pH, non-fermentase, the pH will be 6. Understood? That is the basis of methyl red test. Next is which test? Next is Vogt-Prosker test. Here also, which medium we are using? The same medium we are used for methyl red test, MRVP medium. That means dextrose phosphate broth. Here, we can inoculate the colonies, okay, the positive presumptive colonies into MRVP broth, okay. We can inoculate the positive presumptive colonies into the MRVP broth and incubate for 37 degrees Celsius for 48 hours. Here, we can check whether it contains acetoin in the medium. That is a non-acidic byproduct. That is the non-acidic byproduct. So, here two reagents. That is baric reagent A. That is 40% KOH. Then baric reagent B. That means alpha naphthol in 95% ethanol. Understood? Alpha naphthol in absolute ethanol. That is baric reagent B. So, first we can inoculate the broth with Escherichia coli and kept for incubation at 37 degrees Celsius for 48 hours. And the next day we can add baric reagent A and B into the broth. Okay, and add the baric reagent A and B, then allow the tubes to stand for 15 minutes before reading the result. That is very important for this test. Okay, the development of cherry red color. The development of cherry red color. This is right. Okay, cherry red color is considered as positive for Vogt-Prosker test. That means it contains acetoin. Okay. It contains acetoin. Acetoin is a precursor. The E. coli is unable to produce acetoin and hence it gives negative result. E. coli is negative for VP test. Okay. E. coli is negative. Okay. E. coli is unable to produce acetyl methyl carbinol. Hence The result is negative. Understood? This is the basis of Voss-Brosker test. Understood? Which is the next test? IMViC. Last is C. I we said that small letter I is for ease of pronunciation. So C is the last one, that is citrate. Here the medium is Simmon citrate agar. Okay, it is Simmon citrate agar, green in color. It contains citrate as a carbon source. Okay, it contains citrate. Which one? The Simmon citrate agar, as the name, from the name you can understand, it contains citrate. Citrate as a carbon source. Okay, we can just streak here. This is slant. Okay, Simmon citrate. This citrate test is always doing in a slant. Okay, for here like this. This is slant. Okay, so here we can streak the inoculate the Escherichia coli. E. coli. Inoculate the Escherichia coli. Okay, then incubate for 37 degrees Celsius for 48 hours. If it is negative, it will be appear as the same. Okay, same as the uninoculated medium. This is negative result. So if it is utilized, if the bacterium present in the medium if it can utilize the citrate present in the medium, that means if it utilizes the medium, it turns into blue color. That means positive. The test is positive. If it turns into blue color, that means positive result. Understood? The Escherichia coli is negative for. So, what is the result of Escherichia coli? Escherichia coli is negative for citrate test. Here, this color is known as deep Prussian blue color. Deep Prussian blue color, that is positive for citrate utilization test. And Escherichia coli is unable to utilize sodium citrate as carbon source. Hence, Escherichia coli gives negative test. Okay. The bacteria, most of the bacteria contains citrate permease. Citrate permease. Okay, the citrate permease present in the bacteria can utilize the citrate present in the medium to produce which acid? Pyruvic acid and carbon dioxide. Understood? If the bacteria contains citrate permease, it utilizes the citrate present on the medium. Understood? The E. coli doesn't contain citrate permease. That's why E. coli gives negative for this test. Understood? So, if the bacteria positive, that means it can, if the bacteria contains this citrate permease, it convert. That means it utilizes the citrate present on the medium, and what are the byproduct? Pyruvic acid and carbon dioxide. Okay. The carbon dioxide combines with sodium. Okay, sodium present in the medium, sodium and water to produce carbon dioxide plus sodium plus water, it produces sodium carbonate. This sodium carbonate will make the medium into alkaline. Understood? Alkaline. Here, this Simmon citrate contains a special pH indicator. The Simmon citrate agar contains bromothymol blue. Okay, bromothymol blue. That means if the bacteria utilize the citrate and produce carbon dioxide, that carbon dioxide will combine with sodium to form sodium carbonate and it make the medium alkaline. Then the pH indicator bromothymol blue will turn into blue in color. Okay, bromothymol blue turn into blue in color. Here, okay. Here we are always doing in slant. For here oxidation is happening. Oxidation. That's why. Okay. That's why slant. Okay. Oxygen is required for the conversion of citrate into pyruvic acid. That's why we always perform this test in slant. So this is the biochemical test for Escherichia coli. Which are the 4 tests? So if we get any water sample, first we perform MBN. Then we will get, after the MBN, the last test, complete test, we will get colonies on the plate. Okay, if we are doing MBN or which test, membrane filter technique, both the end result is plate with colonies. Then what we need to do? We need to characterize the colonies. We know that may be Escherichia coli, but we need to confirm it by doing biochemical test. The biochemical test for Escherichia coli is IMViC test. Indole, methyl red, Vox Prosker, and last one, citrate test. And this is for biochemical characterization of Escherichia coli. Okay. Anyway, this was a previous question for CSIR. Okay. So this is for important for junior scientific assistant and for CSIR point of view. Okay. Junior scientific assistant, what you need to study? You need to study the name of this test. What is citrate test is for Escherichia coli. Escherichia coli will appear as, what is the result for citrate test? That is negative. Okay. Then VP also negative. Then E. coli is positive for indole and MRVP. Likewise, you need to study. You need to study the medium for citratus, then medium for MRVP, okay, medium for indole, all the basics. Okay, you need to study a little, but basics. For CSIR point of view, you need to study everything. Okay. So if you like this video, please like and share. Okay, sharing is important. Okay. So thank you. We will see you in the next video. Thank you.